putting in common. Good afternoon, one and all. So now we can start the session. So today's lecture will be about the interaction, or otherwise the nanoparticle interaction with microbes in the environment. Before that, slide is visible. Is this one? I hope the slides are visible.
Yes, sir. Yes, sir.
Yeah. Okay. Just a minute. So good afternoon, one and all. Today we are here to I will take you through you all towards the interaction of these nanoparticle, especially with respect to the microbes present in wastewater. So in the previous sessions, especially Dr. Pokhriyal's session, we could be able to listen about the nanotoxicology, the ecotoxicology of the nanoparticles, which are there, and then in continuation with that, also he has also done similar kind of work, more or less the same, or what not more or less the same, but with the different nanoparticles with coating and then everything. But so let us look into how these nanoparticles behave with respect to the microbes which are present in the wastewater environment. So here the microbes which I mentioned is in the wastewater environment. So this is again the generic picture. Again, it is nothing but the roots of nanoparticles, the pathways, or otherwise the route you can say, or the pathways of nanoparticles in the environment. So these nanoparticles, be it the man-made or anthropogenic, or otherwise natural or incidental nanoparticles, at last reach the first step. It reaches the water or wastewater stream, and then because of the biosolids application to the agricultural field, and then the treatment of wastewater, treated wastewater being let into the natural surface water bodies, at last they enters the life chain of the humans. Not only the life chain of the humans, but also the terrestrial animals as well as the plants. The terrestrial environment it involves. So the man-made or these man-made products or the contaminants, we can say that these emerging contaminants, once their presence is there or over in the environment, they somehow or the other get onto the terrestrial environment, and then they keep impacting the environment in one or other way. And then also they pose, as the studies are picking up, and then there are several studies to quantify what is the risk that they possess when it has been taken as a in the form of food. So with respect to the pathways, there are different pathways. So be it this image, I think we can able to correlate easily, where this nanoparticle coated socks material, once it has been washed, at last they end up in wastewater treatment plants, and then they at last these treated wastewater can get onto the sludge as well as to the supernatant? What if this sludge as well as the supernatant is being used for agricultural fields to cultivate the plants, and then what is the risk that is posed to the humans especially? So the ENPs is nothing but the engineered nanoparticles released onto the environment, be it the soluble nanoparticles or the insoluble ones. So all pose a severe threat to the environment, and then they keep changing as in one or the nanoparticle form or the ionic form. They pose a severe threat to the treatment system as well as the environment. So when it comes to toxicity towards the bacteria, so we have also seen, I think in another way, so how this parameters, especially in water as well as the wastewater, play a vital role in imparting toxicity towards the bacteria. So be it the nanoparticles, these nanoparticles, either it can be single nanoparticle and or mixture of nanoparticles. In a lab scale study, the single nanoparticle case will be taken, and then it will be discussed in detail. But when it comes to the wastewater treatment system, or otherwise the wastewater conveyance or distribution system, wastewater collection system, so mixture of nanoparticles will be there in the stream, and how do they behave? So that is one such question that that needs to be understood well enough. Then only the impact or the toxicity towards of these nanoparticles to the microbes can be well understood. So these nanoparticles of different size would be there, shape would be there, amorphous structure, crystalline nature would be there, and then based upon their size, already in the last 2 days we have seen that how this is colloidal parameters, or otherwise the colloidal properties, otherwise the physical chemical properties of these nanoparticle influence in release of metal ions from the nanoparticles, or otherwise the ions, and then how this size enters into the microbes and then ruptures the cell wall, and then damages the cell wall, so that at last the microbe itself or the bacteria itself gets dead. So if you look at the literature, the size of nanoparticles, if it is less than 50 nanometers, they are more prevalent in damaging the cell wall of the bacteria. And then again, if they are less than 50 nanometer, they release metal ions. Still, those metal ions pose a severe threat to the existence of these microbes, so or the bacteria. So the size plays a vital role. And then assume that if the particles are greater than in size, so it is greater than 50 nanometer in size, how do they behave? So that is one such thing. So in the yesterday's class, I think we have also seen that these nanoparticles, if it is again greater than 50 nanometers, they get aggregated. So either the nanoparticle physicochemical or transformation that we have also seen, physical transformation, chemical transformation, biological transformation. So in this, so either the particle can get agglomerated and then they can get settled. So once it is that is the case, then the water quality parameters play a vital role. So be it the multivalent cations or the natural available organic matter, the ionic strength. So again, where the sand dunes formation, where I gave an example, the pH of the solution. How does this ionic pH plays a vital role in imparting toxicity of these nanoparticles towards the microbes. So all these things play a vital role in imparting toxicity or reducing the toxicity, you can say, towards the bacteria by aggregation. So in the last session of mine, so again I said that when compared to the surface area of these small particles to the bigger particles, the reactivity will be much more high with respect to the smaller particles, whereas when compared to the larger particles. So again, these large particles or aggregated particles settles down and then it gets absorbed or otherwise onto the sludge. It sits onto the sludge or gets carried away with respect to the sludge. So this sludge, when applied to the fertile lands, and then there is a possibility. Are there a possibility is one such question. If there is a possibility, and then what happens to these nanoparticles containing sludge? So these nanoparticle containing sludge can either settle out in the soil. They can again interfere in the bacteria that are present in the soil, or it can be uptaken in any of the plants which has been cultivated with the treated water or otherwise with the groundwater, be it whatever be the case. With respect to the other things. If it is being uptaken by the plants, again it gets into the food chain or otherwise the food cycle. So again, that is very difficult to track the pathways or otherwise each and every step or every movement of these nanoparticles, either in agricultural lands or in biosolids or in the plants. And then with respect to the stabilized or nanoparticle or with the interference of any of the biomass, EPS, again, extracellular polymeric substances also induces uh, particles to remain stable, and then again they can get absorbed onto the surface, and then they don't in that way they can also get help them to remove from the system. So again, when they escape from the system, what happens to that? So what is their concentration? That is one such major thing. So whether the concentration that gets escaped is sufficient enough, or otherwise is it fine enough so that it doesn't pose a severe threat to the environment outside, or does it pose a severe threat? So again, everything is depends upon the concentration of these nanoparticles in wastewater. So when it comes to the concentration, it will not be single nanoparticle. Even in this session, today's morning session, even Professor Lok was also saying that the mixture toxic effect has not been still well understood or well studied by the researchers across the scientific community. And then because wastewater is once it's heterogeneous mixture and then a heterogeneous nature, so these nanoparticles which are there in one or the other form, or otherwise nanocomposites or metal nanoparticles or organic nanoparticles or carbon nanotubes, how do they mingle, how do they react in the wastewater stream is one such question. So again, what is their effect onto the biological functioning of the microbes? So that is one such question that needs to be answered or wanted to be answered. So are interested to know by each and every researcher. So nanotoxicity is nothing but the toxic effect of nanoparticle on biological system and environment. Why, as a researcher or an environmental engineer, we need to care about? Because these nanoparticles can be found, or otherwise they are available everywhere. Rather than having can be found in everywhere, they are available everywhere. They have been incorporated in different products, and then they have been used in different products, so that either it can get through ingestion or inhalation or absorption onto the surface, or if incorporated into other organisms, it may enter the food chain. That is what I was saying, that if it is incorporated into the growth of a plant, and then it may possibly enter into the food chain. So regulations haven't not been established for labeling all nanoparticles. So that is one such thing. Even for the Professor Lokes, in one of the slides, he did mention that that these nanoparticles are the nanoparticles that are used in the consumer product is still not documented yet. So if those are not being documented or popularized among the community, so again, the regulations in determining these nanoparticles, which is there in wastewater, and quantifying them and then arriving at a permissible limit is one such a big thing that needs to be done, or it needs to be focused. And then health concern, accumulation in tissues. So bioaccumulation, otherwise these particles keeps getting accumulated onto the tissues of the microbes, otherwise the bacteria will leaves them or poses a severe threat to the bacteria itself. So nanoparticle in wastewater, most of the studies in around 5 or 6 years back or 7 years back, there was not much of studies on what is the concentration. Then and there were one such group working with Arizona State University, where they were literally identifying the pathways of titanium dioxide nanoparticles in wastewater stream, and then they found out how does it, or what is the fate, or how does it behave in the wastewater environment, whether it gets onto the, whether it is there in the stable medium, or it gets suspended, or otherwise gets aggregated, it escapes from the effluent, or it gets onto the biosolids or solids, suspended solids. So with respect to this particle size, if you could be able to see that this is one such study that has been done in Germany, where quantification of nanoscale silver particles were done and released from municipal wastewater treatment plants as well. So when you look at it, ZM and ZH are nothing but the locations, and then these are the two different nanoparticles that were looked upon in the wastewater. So when you look over the period of time, the particle size range was in between. It is not less than 100 nanometers, but they were there in the aggregated. Already they were there in the aggregated form. So they are about to settle in the wastewater. So again, if these gets bigger and bigger, obviously these 200 nanometers grows out to be 600 or 800 or 1000. Obviously these nanoparticles will settle down into the sludge, and then there are less chances of these nanoparticles getting escaped from the effluent system. So again, when it escapes, so again, what happens? So in wastewater, again, as it is evident, it is also evident that these nanoparticles are there in the wastewater system. How do they behave? So this is one such a big thing that how do they interact? How do they behave is one such big question that is being asked. And one such thing is being said is that in this study, they took zinc oxide nanoparticles and then they looked into how do they behave with respect to the activated sludge. So here, what they did was in this study, they had looked into the concentration of 10 and 50 milligram per liter of zinc oxide nanoparticles. And then they looked into how do they behave, or how do they manage, or how do they react with respect to the nutrients which are present in the, or otherwise the nutrial removal in the wastewater system. So in that sense, so again, this was one such thing that was looked into, and then whether they enhance the they enhance or otherwise they deteriorate the removal of nutrient removal from this treatment system was looked into. And then it was found that inhibition of nitrogen as well as phosphorus removal induced by high concentration of these nanoparticles was mainly due to the release of the metal ions from these nanoparticles and increase of ROS, that is nothing but reactive oxygen species production, which caused mainly the inhibitory action towards these bacteria related to nitrogen and phosphorus removal. So one such thing is that metal ion release from this metal nanoparticle. Apart from that, with the help of sunlight, they generate ROS, reactive oxygen species. So one such thing is that, so these two, now it is very clear or evident that these play not only the size as well as the shape play a vital role, but again the light also plays a vital role, and then which helps in generating the reactive oxygen species. So just a minute, I'll stop sharing. Some disturbance keep coming. So moving on. Yeah. So moving on, with respect to that's what we're discussing, that so these nanoparticles which are there present in the wastewater, they also they release, because of the release of metal ions and then the generation of reactive oxygen species, they had a great impact on the inhibition of nitrogen and phosphorus removal from the wastewater system itself. So though it is not visible, or though it is not visible to the human eyes, but still they have a great impact onto the removal of the nutrients in wastewater system. So when it comes to wastewater, how long does it stay? So that is one such thing. Whether these nanoparticle has a prolonged effect on towards the bacteria is one such question, or these bacteria or these nanoparticles doesn't have any impact on towards the microorganism. So what is the short term as well as long term, whether they keep having their impact over the period of time when study, say assume that it is 45 days or 30 days or 60 days or 180 days or 270 or 365. So what is their impact, long term impact? How do they behave when these microbes have been exposed to the nanoparticles continuously, and then what happens when they have been spiked, that is nothing but given a shock a concentration of these nanoparticles in wastewater? Do they behave similarly, or do they behave completely in a different manner? So let us look into that. So let us take this as a case study where two different nanoparticles has been taken, and then one such thing, the other combination was supposed to be the mixture of these two nanoparticles. So obviously silver oxide and titanium dioxide. Silver oxide, the oxidized form, the most commonly used antibacterial nanoparticle across all the domains, and titanium dioxide is one such material that has been vastly used apart from silver, zinc, copper nanoparticle. So mixture was replicated or studied in order to know the or otherwise mimic the real-time scenario what happens inside the wastewater treatment system. So the three different concentrations were looked into. So there is 0.1, 1, and 10 milligram per liter. And then the solution concentration, otherwise the solution characteristics was also changed. pH of the solution, that was changed to 5 and 10. And then ionic strength, two different ionic strength concentrations were also used in order to understand what would be the impact of these three nanoparticles, be it the silver, titanium, silver oxide, titanium dioxide, and the mixture of these two of three different concentration onto a wastewater. So 2-3 parameters were measured for a shorter duration of time. That is nothing but what is the uptake, oxygen uptake by the microorganisms, and then that is a BOD5 reduction as well as COD reduction. What happens when this has been spiked, and then what happens to these microorganisms. So when it comes to so percentage of specific oxygen uptake reduction, so so with respect to different pH conditions, so be it this 7 or 5 and 10 were looked into, and then with respect to these three, the blank one denotes silver, and then the slightly hazed one denotes titanium, and then the bars with diagonal diagonal diagram is nothing but the mixture of these two nanoparticles. So when this specific uptake rate was, yeah, is there anything? Yes. So at different pH conditions, how does these nanoparticles behave? So with respect to the nanoparticle concentration, it was evident that in order to arrive at the toxic effect, so it is better to use the greater concentration of these nanoparticles towards the microbe. So 0.1 didn't had, otherwise it had the impact, but still it was very difficult to vary with respect to the single versus the mixture of nanoparticles. But at higher concentration, mixture of nanoparticle was found to be dominant when compared to the single nanoparticles. So that is the case for a neutral pH, but it was the case for all the pH conditions that were looked at when studied at 0 molar ionic strength. So, and then again, one such attempt was made. How does these nanoparticle, okay, at 0 molar ionic strength, if there is in such a way that if no ionic strength concentration or the electrolyte concentration is not there, then we could able to arrive at this mixture had a greater impact than the single nanoparticles. But when the concentration was increased to 0.1 molar of ionic strength, then again in cases where in some of the cases the single nanoparticles had a greater say than the mixture of nanoparticles. So this states that with respect to this, it's clear that with a shorter span of time, the characteristics of the water and then the constituents that is being present in the water plays a vital role in imparting toxicity or having an impact on towards the wastewater treatment. So moving on, so again, one such thing was again BOD reduction was also done. So in similar fashion with respect to the specific oxygen uptake rate. So again, a mixture had a greater say with respect to 0.0 molar ionic strength, and then with respect to acidic pH, again titanium and mixture had a more or less same kind of effect on towards the reduction pattern, and then whereas for alkaline pH conditions, so there were greater impact by the mixture of nanoparticles than the single nanoparticles. So with respect to point one, so as like the SOR, similar kind of results were expected where the single nanoparticles at higher ionic strength outplayed the mixture toxic effect of nanoparticles. So with respect to percentage COD reduction, again, these nanoparticles were spiked onto the solution, and then the impact of these nanoparticles onto the solution was also determined. So in that case, what would be, because when compared with the BOD, COD is one such experiment where you would be able to arrive at the results in a very shorter duration of time. So again, closed reflex method was used, and then it was found that among nanoparticles, with respect to the percentage removal of COD with respect to time, as this was for 5 years after the nanoparticles was spiked into the solution. With respect among the nanoparticles, titanium had the higher impact, or otherwise outplayed the other two nanoparticles, that is nothing but silver oxide as well as mixture of nanoparticles. So from these short-term studies, we could able to arrive at, so though they have, or otherwise though they are there in the solution, they do have significant impact on towards the reduction of be it the BOD or the nutrient removal in the system. So moving on, taking it after looking into different literatures, so one such attempt was made in order to have an long-term impact of these nanoparticles onto the system. So in that sense, activate aerobic reactor was set up at a lab scale, and then the influent concentration, or otherwise the load that was given to be was 500 milligram per liter, and then two different concentrations. So C mentions here the control where no such nanoparticle was spiked in. 1 and 10 is nothing but milligram per liter, or the concentration of these nanoparticles, and then around 225 days was the period of studies. Around 180 days was the study period where these nanoparticles was spiked, and their impact on towards the sludge as well as the supernatant was continuously monitored for each and every. So when it comes with respect to the concentration of these metal ions in wastewater, be it in sludge or otherwise in supernatant, with respect to silver, the silver concentration was found to be very high in the supernatant. And then also because as silver already discussed, they can also combine with the ligands which are being present in the wastewater. So that is one such thing is that chloride, again, silver combines with chloride to form silver chloride. And then there were evidences that the white precipitate at the bottom of the reactor is nothing but the silver chloride because of this reaction. And then because of this, silver ion, or otherwise the silver chloride, which gets onto the sludge, or when the sludge has been taken, so the greater concentration was more or less because of this event that happened inside the reactor. So when exposed to 1 milligram per liter, the maximum COD reduction was found to be 83.5 after 105 days, whereas for 10 milligram per liter, after 45 days of exposure, it was around 81%. So that is the maximum reduction in COD that was observed. And then with respect to the TEM or EDX image, so the sludge sample, when it was analyzed, it could able to get from the sludge that silver was found in the sludge. So apart from that, the other picture was also not there here. So again, silver as well as chloride was also looked into whether their presence is available in the sludge or not. So similar kind of effort was made with respect to titanium dioxide for a long-term study, and then here similar kind of analysis were done, where the nanoparticles was exposed towards the microbes were exposed towards the nanoparticles, and then at two different concentration, that is 1 and 10 milligram per liter. And then here, with respect to the supernatant, the titanium concentration was not that high because of their insoluble nature, but at the same time, their concentration in the sludge was pretty much high than the silver nanoparticles. So with respect to the concentration, that is nothing but 1 milligram per liter. When exposed, the percentage reduction in COD was found to be around 83%, which was after 105 days. And then again, after for 10 milligram per liter, it was found to be around after 75 days, or 50 days of exposure. So similar kind of analysis again with respect to titanium dioxide, when analyzed with respect to TEM as well as SEM, it was found out that these nanoparticles started getting aggregated over the period of time. So this was nothing but a 90-day period and then 180-day period. How do they look is one such thing under the TEM, and then EDX was also analyzed, and then the confirmation of though there are different constituents that are being present in the sludge, but titanium was found to be the dominant in sludge when analyzed. So similar kind of study was also made with respect to the mixture of nanoparticle. What is the combination that it plays, and then how does its impact the microbes? So two nanoparticles were mixed at two different concentrations, at 1 milligram per liter as well as 10 milligram per liter, and then similar kind of analysis were made. And then again, with respect to the other two nanoparticles, a mixture had a greater impact onto the reduction of COD than the single nanoparticles. So again, mixture had the higher say than the single nanoparticles. So again, moving on with respect to the results that we could able to arrive at, it was the silver chloride precipitate was found along with the sludge when the nanoparticles were exposed into the reactor at two different concentrations. And then with respect to the metal ion concentration, it was greater than in sludge than in supernatant. Those are the reasons why it is very high. And then affinity, when it comes to affinity or the closeness of the sludge towards the nanoparticle, titanium had a greater affinity than the silver nanoparticles. And then with respect to COD reduction, mixture toxicity of the nanoparticles was found to be predominantly high than the individual nanoparticles which is being present. When we looked upon the antagonistic as well as synergistic effect, the antagonistic effect was observed. That is nothing but the summation of the individual nanoparticles in the wastewater outperformed the mixture toxic effect on towards the COD reduction. So be it for both the concentration, it outperformed. So this was one such study that was done during my research work, and then similar kind of analysis was also being done erstwhile in the year 2008. This was one such early study that where it was evident that these nanoparticles gets onto the sludge. So these nanoparticles, when they either it can be there getting absorbed onto the medium or get entrapped or gets aggregated. Because of its aggregation, it can get settled over, and then complexation again, it forms a mass and then it gets settled out, or it degrades. There are very less chance for these nanoparticles to get degraded. And then the reactivity, whether they produce the reactive oxygen species, so that these target the cell wall of these microbes, and then they deteriorate their health. And mobility into other environmental components. So when these two combine, how do they move or how do they behave is one such question. And then this is one such this is Limbach is one such early researcher, and then where they found the nanoparticles getting stick onto the sludge. So moving on. So with respect to the impact on removal mechanism of or otherwise the impact of these nanoparticles onto the sludge was evident from long-term and short-term studies. The one such study also was looked into for the germination of seeds. If this germination of seeds was used to have this nanoparticle containing sample or concentration, how does this concentration of these nanoparticles retards the growth of these seeds which gets germinated. So one such thing, control, is nothing but the longer one without the exposure, whereas a 10 denotes the concentration. CUO is nothing but the nanoparticle that has been chosen was for consideration. 100 is nothing but the concentration. 1000. So when you look at it, so greater the concentration, there were greater chance of retardation of growth. So again, so one such study was looked into what happens when this the sludge which had the nanoparticles gets onto the root system, and do they really pose a severe threat on towards the humans. So in that case, one such attempt was really made where the sludge was mixed with nanoparticles, and then it was exposed to leafy plants. So and then it was checked what would be the concentration of these nanoparticles on root, shoot, and then as well as their photosynthetic property using chlorophyll A and chlorophyll B analysis. So nanoparticle exposure towards a cell. One such pathway is nothing but the disruption of cell wall and plasma membrane components. So smaller the size, greater the chances of cell wall disruption. Decrease in ATP adenine triphosphate level and collapse of plasma membrane. So that is one such mode of way in which the nanoparticles enter and then rupture the cell wall and then start doing the damage. Induction of pits and gaps in cell wall. So this also leads to the damage of the cell. Accumulation in cell wall and decreases its permeability. So what these nanoparticles adhere to the surface of the microbes, and then they start damaging the cell microbe as such. Increased ion permeability of the cell membrane and inhibition of cell wall synthesis. So again, these two also plays a vital role. And then here I haven't mentioned about the production of ROS. So again, the reactive oxidation species, how do they get generated, and then whether they rupture the cell wall, and then because of that, these nanoparticles gets into the cell wall and then start deteriorating. So this is one such study where with before the treatment of nanoparticle and after the treatment of silver nanoparticle, they could able to have a clear or distinct effect towards the bacterial cells. So before treatment, A is nothing but they have a very condensed way of cell wall, and then B2F is nothing but after exposure to silver nanoparticle, over the period of time, the cell itself got disintegrated, and then there is no life in that cell. So these nanoparticles stick onto the wall, rupture the wall, or form pits, or get absorbed, and then start deteriorating the cell. So when it comes to toxicity assessment of these nanoparticles, so as I mentioned that one such attempt was made in order to arrive at the risk that these nanoparticle poses to the humans. So one such attempt was made where these nanoparticles was mixed with sludge and then irrigated to a plant. After 12 weeks, again the plant was taken for analysis, where their root, shoot, as well as the leaves were taken into consideration for their metal ion concentration, and then it was. And then this was also one such study where when they had the nanoparticle being exposed to fish, how does this nanoparticle settles onto their layer, and then how do they venture into them is once a pictorial it is being represented. So actually, with respect to the living creatures in India, there is not much of studies that have been gone in, but still people in the West have really explored a lot with respect to this nanoparticle, getting how it gets absorbed onto it and then how it keeps moving inside the system. So with respect to the interaction as well as the risk it possesses towards the microbes, so there are two types of risks. One is the health risk as well as the ecological risk. For risk, there needs to be three components. One is the source, one is the medium, and one is the receptor. So the source is nothing but the source of generation, or where the particles or the nanoparticles itself is, and then medium in which the material gets transported, be it water or soil or air, so in which it gets transported or moving. Receptor is nothing but the targeted species, so be it the human or otherwise the aquatic species or the microbes or otherwise the environment as such. So with respect to water, it is nothing but the ingestion. So we drink this nanoparticle containing water, what is the risk that it possesses? Air is nothing but the inhalation. If we respire, the soot that comes out from burning, that is one such thing. And soil, again, ingestion. The root is nothing but the plants that are cultivated using the contaminated soil. So how does that get ingested through the mouth? So those are the pathways. So there are several terminologies. One is I think this has also been touched up by Professor Dubey, but still I will just have a quick overview of this. Background, incremental and total risk. Background risk is nothing but the risk what people is exposed to from a given medium, be it the water, soil or air. Incremental risk is nothing but due to the addition of external pollutant. So assume that this nanoparticle is nothing but an external pollutant. What is the risk because of this nanoparticle? Total risk is nothing but the background as well as the incremental risk, and acceptable risk is nothing but the allowable risk for a particular contaminant can result in only it on a receptor. So it is nothing but the total divided by the risk ratio can be given as the total risk is nothing but the background plus incremental divided by the acceptable risk. So that gives you a ratio. If it is false less than 1, then it is of no concern, and then if it is of greater than 1, then it is of real concern. So this is a small example. I think this you can look into it, where in US the excess lifetime risk of getting a cancer is 10 power minus 6, that is the acceptable risk, and background risk of getting a cancer is say 10 power minus 5. So calculate total risk of getting a cancer and calculate the risk ratio. So here the incremental risk of getting cancer is 10 power 5. So the background risk again is a 10 power the incremental risk is nothing but 10 power minus 6, and then background risk is nothing but 10 power 5. The total risk comes out to be 10 power 1.1 into 10 power minus 5. Whereas when you calculate the risk ratio, so that is nothing but the acceptable total divided by the acceptable. So it is greater than 1, and then that means that it is of real concern. So so similar kind of study was also done. Just I will go through it. So whereas we're mentioning these nanoparticle different products, once it gets onto the sludge, so this sludge, this pot was exposed to two different nanoparticles, and then what happens with respect to the concentration in leaf as well as shoot. So I'll just take down towards that. Yeah, this is the slide where two different this equation was used. Di is nothing but the dietary intake, A is nothing but the metal concentration in spinach leaves, and then that is nothing that has been given as milligram per kilogram of fresh weight. B represents intake of spinach per serving. So how much amount of serving per person per day? So it can be in grams or in kilograms. C represents the number of serving, the number of times that the food has been taken, and D represents the body weight. So with respect to body weight, two different concentrations were given, silver as well as mixture plus silver. So with respect to that, so for the infants, they with respect to the body weight, they had a severe, they were crossing the dietary reference mark, and then they really possesses a risk towards the humans. So consumption of spinach shoots from plants which have been exposed to a concentration of say 10 milligram of nanoparticles per kilogram of sludge, resulting in the dietary intake estimates of greater than. So that means that there is a higher reference dose for all age mass class for both silver as well as titanium dioxide nanoparticles. So also it has been also concluded that, yeah. Yes. Okay. So further, it was also concluded that the consumption of spinach shoots of plants exposed to mixture of nanoparticles, say assume 10 milligram of nanoparticles per kilogram of sludge, resulting in dietary intake estimates higher than the reference dose of less than 50 kilogram of body mass. So the lesser the body mass, and then there was a greater risk. And then, so this is one such study that conveyed that these nanoparticles possesses, once they enter the food chain, they possesses a severe threat towards the humans as well as the environment. So how do they behave in the soil is one such thing. So whether they are there in the ionic form or they are there in the nanoparticle as such, uh, uh, whether they release the metal ions or whether they stick onto the soil and then they affect the bacteria which are present in the soil is again one such question that we need to look in detail in order to arrive, or otherwise in order to conclude how do they interact with respect to the microbes. So with respect to risk assessment, these are the 4 initial steps that we need to follow. That is nothing but the hazard identification. Hazard identification is nothing but the nanoparticles or otherwise any emerging pollutant that comes in contact. We can say it as the hazard, and then how it is being identified in the environment, and then exposure assessment and dose response. So what kind of exposure and then what kind of dose that we are going to give, and then based upon that we interpret and then characterize the risk. So here once you characterize the risk, if it is less than 1 then it is of no concern, and then if it is of greater than 1 then it is of real concern. So this is how we could able to carry out a study, and then we can able to correlate their impact on towards the wastewater as well as on towards the water. With respect to water, once they escape the effluent, again a detailed analysis can be done with respect to the water that you pick it up, and then several stability studies or sedimentation analysis can also be done, or otherwise a modeling can also be taken up in order to arrive at how these nanoparticles behave in the water. So different types of water can be picked up. So that is one such thing. The pH as well as the ionic strength concentration can be adjusted, and then with the help of dynamic light scattering instrument, you can technique or otherwise DLS, you can able to arrive at the settling pattern of these nanoparticles in water. So once the settling pattern of these nanoparticles gets clear, so at what rate these nanoparticles settles out from the solution, or are they stable enough? So if they are stable enough, what kind of coagulant that needs to be there in place in order to remove them is one such question. So again, those kind of studies can also be done, and then that will help really in order to arrive at how do they react, or how do they react in water. So rather than having it in wastewater, how do they react in water again is one such topic of concern. So again, that when it is there in water, whether it is there in continuous motion or it gets settled. So again, when it gets settled, again there is a chance of settling in soil. What happens to that is again one such question that needs to be looked in detail. So with respect to be it the detection in wastewater or the detection in water, so there are several challenges in order to arrive or be specific with respect to the nanoparticles which are there in water as well as wastewater. So those quantification techniques we will be having a look in the upcoming session. So that is in tomorrow's session. So that will help us to have a combination of 2-3 instruments or otherwise analysis that will help us to reach out the exact concentration as well as the size of nanoparticles in the medium that we are looking for. So with this, I will stop for today's session. So if there are any queries, then you can drop in. question. Okay, why in short-term exposure, individual nanoparticle titanium had more impact on organic matter to move, whereas in long-term mixture. Okay, with respect to short-term, see, these nanoparticle might have, see, with respect to short-term, the study was done for 5-hour duration. This nanoparticle was spiked, and then for each and every hour, the report was taken. With respect to the long-term impact, the nanoparticle, once the reactor was stabilized, and then the nanoparticle was continuously being fed into the reactor, and then the concentration of the a little bit amount of sludge will also be taken in order to maintain the reactor. So in that case, there might be the chance of having a greater impact of these nanoparticles at the initial stage, but when compared to the other nanoparticle, because of the release of metal ions, because of the silver, so the combination of these two nanoparticles might have resulted in greater impact on towards the microbe rather than the single nanoparticle. Is that okay? Yeah, okay. Okay, so if there are any queries, still they can drop it in chat box. Can have a small discussion.
Yes, sir. While looking into the individual nanoparticle also, we had more impact because of TiO2 than Ag. Is there any reason for that?
Come back again with respect to individual nanoparticle. Why
Yes,
there
sir.
is a impact of silver, greater impact of titanium than silver, right?
Yes, sir. Yes, sir.
See, though silver initially had, if you look at the thing, those silver initially had the impact on towards the microbe, but the rate at which the silver gets converted to silver chloride was very high when compared with the titanium. So titanium was insoluble, and then again, because the addition or the accumulation of this titanium outperformed the results of silver. That is what I can conclude. Yeah, I hope I answered your query.
Yes, sir. Thanks.
Okay, so if that's the case, so then we will meet up for tomorrow's session. So that will start again at 9 o'clock, where Professor Lok will be taking us forward with this session. So tomorrow, 2 sessions will be of Professor Lok's, 9 to 11, and then afternoon session I will be dealing. So thank you all for joining. So let us meet. If there are no queries, then we will wind up the session, and then we will meet tomorrow morning at sharp at 8:55. Thank you.